Hello, my name is Klaus Eckelt and I'm a PhD student and doctoral researcher at the Johannes Kepler University in Linz. It's my pleasure to talk about the work that I've done together with my colleagues, and we form the Visual Data Science Lab at the Johannes Kepler University in Linz. Over the past year, we worked on the visual exploration of relationships and structure in low-dimensional embeddings, and this was also recently accepted as a paper in the IEEE TVCG journal. The initial motivation for that was that we had patient cohorts defined by certain features like tumor type and specific gene mutations, splitting the data in multiple subsets of interest. And for these subsets, we wanted to get an overview over the high-dimensional data, specifically their genomic data, such as gene mutations and gene expression. But there are lots of these attributes, potentially hundreds or even thousands of them. So we applied dimensionality reduction algorithms to the data and tried to track whether the cohorts diverge in the projected space through our operations. But this became challenging quickly using standard layered enrichment techniques such as color encoding or by using different shapes. While mutually exclusive cohorts are easily represented, as with color here, it becomes increasingly harder to depict items that belong to multiple cohorts. For example, if we split this pink cohort even further, for example, whether or not a specific gene is mutated, we could now introduce either a new color, but then we would lose the relationship to this parent group that both of these belong to, or we can use a new shape to denote this difference in between them. But the number of options that we have to encode additional information is very limited and also gets overwhelming very fast for the users. With more items in the embedding, identifying distribution differences between the cohorts also becomes more challenging. And while the embedding might show differences between the cohorts, there is no explanation on what attributes drive these differences. And that's where we came in with our approaching tool. We integrated our approach for visual exploration of relationships and structure in the Projection Space Explorer, an open source library to calculate and explore scatter plots of projected high-dimensional datasets. We extended the standard scatter plots to also define and visualize groups in it. The screenshot shows an embedding of 3 tumor types, which I'm going to talk about in more detail on the next slides. But first, I want to highlight 2 things. The groups that are shown within the scatter plot, represented by gray diamonds and here additionally labeled, and the sidebar on the right that summarizes groups and allows comparisons of the groups by the high-dimensional data. The approach is very general, and I will mention a couple of other examples we applied it to, but for the purpose of this talk, I will mainly stick to the cancer genomics use case. So what is this use case about? It is based on a New York Times article from 2021 about a new drug to treat cancer patients with a specific gene mutation, namely KRAS G12C. This mutation mostly occurs in lung cancer, but also patients with pancreatic or colon adenocarcinomas are affected. Using data from the Cancer Genome Atlas, we want to explore these 3 tumor types in more detail. And if you would like to repeat the use case yourself, it is also described in our paper, and the dataset is preloaded in our online tool. Let's revisit the scatter plot now there. It shows the 3 tumor types that are represented with color, and additionally we vary the shape of the points to show whether a particular patient has a mutated KRAS gene and therefore might benefit from the drug discussed in the article. While the 3 tumor types are visually separable, it is hard to tell whether the mutated and non-mutated subgroups, which vary by shape, are distributed differently, and there is no easy way to select the subgroups. That's why we thought about how to represent groups in the embedding. For this, I'm going to use a simplified scatter plot to introduce our idea and then get back to the real data afterwards. We can see a scatter plot with similar distributed points using the same layout enrichment as before. And I will now start to identify some groups of interest, first by selecting all of the green items that belong to the lung cancer patients. And we can now represent this selection through a diamond symbol. So the diamond symbols represent our groups, and these are always rendered on top of the scatter plot, so above any other items that we might have there, and they also scale in their size depending on how large the points are in the scatter plot. I can also create additional groups, once for the mutated samples and once for the non-mutated samples. This gives us 3 groups, and 2 of them are subgroups of another, so we can also make this relationship explicit by drawing edges from the parent to the child group. So now we created a structure, namely groups, through selection, and there are different ways to define structure. So first of all, we might have no structure at all, just the single items. Then we would have the same behavior as in the standard scatter plot. But we can create groups through user selection, as we just did. We can also create groups through clustering in the scatter plot. And we can create groups based on the high-dimensional data, for example, the tumor type, creating a group for the lung and the colon cancer patients, or creating a group based on a certain age range of interest for our analysis. And in between these groups and from the items to the groups, we have relationships. So we can have a relationship between groups. There we use directed edges that are drawn also on top of the items in the scatter plot. And they can be extended to arbitrary depths. So we can create very deep hierarchies using this. But we can also create graphs by introducing additional directed edges. We can have bidirectional connections between groups. We can also have circular relationships. And this makes defining structure very flexible. The relationships between items and groups are shown on demand by selecting either an item or a group. Let's start with an item. If we select that item, it gets connected to all the centroids of the groups it belongs to. And if we select a group in turn, then all items that belong to that group are connected to the centroid. The centroid's position is the average position of all its points in the scatter plot. So typically it is placed at the center of all the points, and the edges form a star-like pattern. That's why we call this visualization the star visualization. And here we also have an alternative, which is the contour plot. This has 2 reasons. First of all, if we define hierarchies, then items are connected to multiple centroids, which might lead to overplotting. And also to get an idea of how the points of a group are distributed in the embedding space, the contour plot gives a better idea because it shows where the most dense regions of that group are. We also support item sequences, which are not going to be the focus of this talk today, but they are particularly handy when dealing with data that changes over time. For example, if we have a set of patients and their data is recorded multiple times over the treatment process, then we have multiple items for the same patient, and these are then connected with a line, and we can then observe how the data of a patient changes over time. And we can of course also define groups in these sequences, which would then give us a group sequence. In the paper we used chess as an example for sequential data, and my colleague Andreas Hinterreiter also used this to analyze how neural networks with different learning rates behave in the training process. Now that we have discussed how to define and represent relationships in structure, I would like to talk about the exploration of these and how we can use them to better understand the differences in the embedding. For that, I will go back to our tool and the genomics use case. And here we projected the data of the 3 tumor types by the gene expression of about 700 cancer-causing genes. we can see that there are two large clusters, one for colon cancer samples and one for lung and pancreatic cancer samples. There's also one cluster at the bottom right, which contains all items for which there is no gene expression data. For each tumor type, there's also one overall group and two subgroups containing the samples with and without the KRAS mutation. For colon and lung cancer, these groups are all close together, but not for pancreatic cancer. So let's take a closer look into that. I start with the pancreas samples that have no KRAS mutation. And interestingly, here we have a couple of samples that are actually located in the cluster of the colon tissue samples. So here the expression value is close to that of colon tissue. And when I select the group with the samples with a KRAS mutation, we can see that multiple samples have no expression values, and this drags the centroid towards the bottom right of the embedding and therefore away from the other group centroid. Let's now get back to the initial use case and see which KRAS mutations are most frequent in the three tumor types. For that, I select the three groups and open the comparison sidebar. This sidebar contains a summary visualization in the first column that shows us the tumor type, whether KRAS is mutated, and what specific mutations occur in this group. There is only one tumor type and one type, and all of the samples are mutated, so here we only see the labels. But for the specific KRAS mutation, we get a bar chart and the most frequent category, which is G12C for lung cancer and G12A for colon and pancreatic cancer groups. We can also see from the tooltip that G12A is only the third most common mutation, with about 11% in the lung cancer group. This difference is also reflected by the difference visualization in the next column. We can see that, of course, the tumor type changes by 100% from lung to colon cancer, but we can also see that the frequency of G12C is reduced by over 40% and the frequency of G12A by about 30%. Smaller changes are hidden by default through a threshold, which is adjustable with the slider at the top. This ensures scalability if there are many small differences, so we can still focus on the large differences between the groups. To better understand the positioning of the groups in the scatter plot, we can also compare the groups by the gene expression that was used to create the scatter plot. For this, I changed the attributes that are shown in the summary and difference visualizations by going to the details tab and selecting all gene expressions of the cancer-causing genes as summary attributes. And we now open the comparison side panel again, but first select the three tumor types once more. As we have several hundred of attributes in the summary visualization now, the ranking of attributes comes into play. Attributes in the summary visualization are ranked by the normalized standard deviation to quickly identify the most and least varying attributes. Here we can also see a different visualization, a density plot that has the overall distribution of the data in black and then the distribution of the group in blue. The difference visualization on the right ranks attributes by the difference between two consecutive groups, and we can see here that between the co-located lung and pancreas groups we have most difference between NKX21, GLI1 and A1CF. NKX21 is a lung cancer biomarker, and we can see it's almost zero for the pancreas samples. Between pancreas and colon cancer samples, the highest difference is in MYB, CDX2 and PER1 and GLI1, which are all related to pancreas and colon cancer. In the paper, we discuss multiple summary and difference visualizations that are tailored to specific domains and use cases. For today's use case, we use the generic summary and difference visualization that show the high-dimensional data either for points or items in the scatter plot, which is shown in the top row, or between groups, as it is shown in the bottom row. But in many cases, it makes sense to create domain-specific summary and difference visualizations. In the paper, we have a second use case on analyzing chess game data. And here we show the game state in the summary and difference visualizations. There is also an example of solving attempts for a Rubik's Cube, where we have an unfolded cube that represents our states and of items and groups. And my colleague Christina Hummer analyzed chemical compounds and visualizes the maximum common substructure inside a group or also in between groups. And she also has a dedicated paper on exploring chemical data sets and analyzing machine learning models that are applied to them. In the future, we are also going to work on comparing embeddings. That's actually already possible now, so you can have multiple scatter plots side by side and see how the data is distributed differently or similarly there. And we're also going to improve the comparison of the data such that we can find combinatorial effects of multiple attributes that might differentiate our groups well. And also currently the groups are compared in the order they are selected and not all against all. My colleague Patrick Adelberger is also going to present Coral today. And Coral is a tool to create and analyze cohorts. And naturally, it would make sense to get an overview over the high-dimensional data of the cohorts that are created there using the approach that I have presented. So that's also something we are working on. If you would like to give it a try, we have an overview page that summarizes the various data sets and domains we applied the Projection Space Explorer to. And there's also a link to our online tool that you can use today and where you can also upload your own data. And as I've already mentioned, the library is also open source and you can integrate it in your own applications. I would like to thank our partners, especially from Boehringer Ingelheim, who gave us valuable feedback on the use case I've presented today. And that brings me to the end of my talk. And I can't answer questions in this recorded version, but my contact details are on the slides, and I'm very happy if someone gets in touch for further information. Thank you.